right, excellent. So we're going to go ahead and get started. If anyone has any audio issues or any concerns, you can go ahead and type that into the questions and we'll see if we can troubleshoot as we go along. But for the moment, we're going to assume that everyone can hear us and see our screen. So thank you for joining us this morning. This is Rieta from the Greater Farallones National Marine Sanctuary, and I am really excited that everyone can join us today for our webinar. So this webinar is hosted by our Kelp Recovery Working Group, which is a joint partnership between the Greater Farallones National Marine Sanctuary, the Greater Farallones Association, and the California Department of Fish and Wildlife. And today we're focusing on kelp monitoring methods and technologies for this webinar. And so for the next 2.5 hours, we have some really amazing speakers lined up that are going to be discussing their expertise, their work in different regions, and for our focus region. So we're going to start off by looking at our objectives. Yep, so go ahead and move on to the next one. Perfect. So today our goal is really to learn from our guest speakers, their efforts in other regions in our region to look at how to survey kelp beds using different methods, different technology. We're going to discuss aerial mapping, remote sensing, and the challenges associated with those and ways to address them, and looking at what the implications could be for our region, and what of those methods seem like they may be the most promising and effective, what could be applied in our region to better understand the changing dynamics in our kelp forests. And we hope after our discussion today that we can look at really starting to craft monitoring recommendations based on what we learned today. So this will hopefully be, the knowledge from this webinar will be a hopeful tool or a useful tool for this group to put together those recommendations for the sanctuary and for CDFW. So we'll be taking really detailed notes today on each of these presentations. And the way that the presentations are going to work is each one will be between 10 to 20 minutes long. And we're going to have 5 minutes at the end of each talk for questions. And so we ask that during these 5 minutes, if you can type your questions, you'll see in your webinar software panel, there's an area to type your questions. And in order to keep the flow sort of smooth and things moving along, we will read those questions to the presenter, and the presenter can answer. And then at the end, we're going to make sure that we copy all of those questions so that if any of them are not addressed during those 5 minutes, we will still have an archive of them that we can send to the presenter that they can then answer at a later time or during our discussion at the end. So during the last part of our webinar, we have time for a group discussion. And let's go ahead and look at the agenda. So if it didn't pop up. Okay, perfect. So for the next couple minutes here, we're going to wrap up our introduction. We're going to have Cynthia discuss the update on the state side of things on CDFW here in just a minute. Our first speaker is going to be Rebecca Flores Miller, who is an environmental scientist with the Department of Fish and Wildlife. And she is the CDFW lead for marine algae management, and she's going to be discussing the California Department of Fish and Wildlife aerial kelp surveys. Then we will move on to Tom Bell. He's a postdoctoral researcher with the University of California, Los Angeles, and the University of Alaska. And he will be discussing remote sensing of kelp canopy density along the California coast. We also have Meredith McPherson, who's a third-year ocean sciences PhD student at UC Santa Cruz in the Kudela lab. And she's looking at different a variety of remote sensing technology that can be used in Northern California to look to study kelp. And she'll be discussing how to observe and monitor bull kelp after this large-scale ecological and environmental perturbations on the North Coast. And after Meredith, we're actually going to take a break. We know this is a very long webinar, so we're going to allow a few minutes for a break for folks that don't want to miss any of the talks or any part of the talks. And then we will have Mysira Costa speak. She's a professor at the University of Victoria. She's the coordinator of the Remote Sensing Laboratory in the Department of Geography, and her expertise is in oceanography and ocean remote sensing. And she'll be discussing detection of bull kelp in the Salish Sea using high-resolution satellite imagery. And then last but not least, we will have Helen Berry, a marine ecologist who maps and monitors nearshore habitat for the Washington State Department of Natural Resources. And she'll be going over floating kelp trends and issues in Washington State. So that's our outline of speakers for today. So I wanted to introduce them at the beginning just really quickly because for our transitions, we'll just say who the next speaker is so that we can keep the flow going. And at the end for the discussion, we will ask that if you have questions, you can either type them in the question box or you can raise your hand. You'll see a little hand symbol on there. And if you raise your hand, we will unmute you so that you can verbally ask your question so that we can have more of a discussion. But for those 5 minutes after each speaker, we'll ask if you can type your questions and we'll go ahead and read them out loud. Okay, and next slide. Yep, so this is just to remind us of our working group goal. Coming out of this webinar and out of our meetings, we're looking to develop research, education, and outreach, and management recommendations for the recovery of bull kelp populations along the north central California coastline. And these recommendations will be presented to our Sanctuary Advisory Council in November of 2018 for discussion and approval. So I am going to go ahead and turn it over to Cynthia, who's going to give us an update on the state side of things. So let's go ahead and unmute Cynthia. She's our Kelp working group, Kelp Recovery Working Group co-chair as well, along with Francesca.
All right, well, I'm just going to very briefly give you guys an update on all the interesting things that have happened in May, because there was quite a few interesting things that I think that this working group will be interested to hear about. I'm going to go in chronological order because that's the way my brain works. But at the beginning of May, I was able to attend the International Abalone Symposium in Xiamen, China, and I thought it was really an amazing experience to be interacting with folks on the international, in our international field who are also dealing with the same issues that we are dealing with over here on large-scale urchin barrens and loss of kelp, and thinking about the impacts to ecosystems and to our economic interests. And so I think that what we're doing over here is going to be looked at more broadly than just within our region. And I think that we can learn a lot from what's going on elsewhere as well. So I just wanted to reiterate for you guys that what we're doing and what we're discussing is really important and will have implications for our area and beyond. So what we're doing is really important. The next thing that happened, and I'll let Josh talk about it maybe at the in the discussion, but he was doing a lot of work over the last couple of months to organize a big recreational harvesting event up in Ocean Cove in Sonoma County, and that occurred over Memorial Day weekend. And just to give an update, we were able to work with him and the recreational divers to get a lot of data, and they were hugely successful. They had 100 divers participating over the weekend, and we estimated that they got 7,500 pounds of purple urchins from that cove. It's an area maybe about 5 acres, and that would be approximately 15 commercial dive days. So I think that's a huge effort by the recreational community, and really shows what can be accomplished, and the benefit of using multiple approaches in combination and making use of the resources we have to the best effect possible. So, and then the last thing is that last week we had another working group meeting focused on kelp forest issues in California. This was to start a discussion more statewide and think longer term about kelp ecosystem management for the state, and identifying major threats and thinking about potential impacts of climate change. So I think, and what we need to do to, how that can inform our fisheries management, how that can inform, the restoration of kelp forests. So I think, again, what we're doing here fits in very well with a lot of the work that is happening more broadly, and so I wanted to give you guys that update on other work that we will be informing with our work.
Okay, perfect. Thank you, Cynthia. We are going to go ahead and have Rebecca start her presentation. So we're going to go ahead and pull it up. And Rebecca, I will unmute you here in just a moment. Okay, perfect.
Hi, can you hear me okay?
Yes, we can hear you great.
Oh, great, thank you. So yes, I'm Rebecca Flores-Miller. I'm an environmental scientist with the Department of Fish and Wildlife, California Department of Fish and Wildlife. Thank you for the opportunity to discuss the California Department of Fish and Wildlife's aerial kelp surveys and the opportunity to learn from others in their work. So today I'll be providing some information on our California Department of Fish and Wildlife aerial kelp surveys. If you could please advance the slide. So throughout the presentation, I'm going to try to refer to the California Department of Fish and Wildlife as CDFW. They may slip in and say department. So I'll begin by providing a background on our CDFW aerial kelp surveys and how to access the data. I'll highlight some of the uses of the data set. I'll provide a snapshot of the results of the Northern California surveys, and I'll also provide an overview of the value and the limitations of the data set. Next slide. Thank you. So the California Department of Fish and Wildlife aerial kelp surveys are performed along the mainland from the U.S.-Mexico border north to the Oregon border and offshore of the Channel Islands. The digital images collected have a 2-meter spatial resolution, and they're multispectral with an infrared band to pick up kelp. The images are processed and georeferenced, and they are classified. They're put into mosaics, and they're classified to distinguish the kelp captured on the images. And both giant and bull kelp are surveyed. The first department survey was conducted in 1989 by a contractor for CDFW. There was a 10-year lapse the next survey in 1999. The 1999 survey was a compilation of surveys by CDFW and 2 other contractors. And in 2002, annual surveys were performed in-house until CDFW no longer had the technology or the capacity to continue the surveys, and then began contracting out the surveys in 2008. So although CDFW strives for a coast-wide survey, there's been times when this has not occurred. There's weather, equipment malfunctions, lack of funding that have all affected the imagery collection. And when available, CDFW has been able to obtain data from other organizations. However, that data can be very patchy and limited. Next slide. So before I talk about the availability of the data set, I wanted to direct you to the images you see at the bottom of your screen. So these are from the 2016 Aerial Kelp Survey in San Luis Obispo County. On the left is an image from the camera system, and the right, after some processing, is a depiction of the surface canopy at that time. So these data are made available on the CDFW webpage in the form of GIS shapefiles, as well as on Marine Bios, which is a CDFW marine and coastal data viewer for folks that do not have access to ArcMap. The data are available by year, and they contain the metadata, which details how and when the surveys were performed, if there were any areas that were missed during the survey, and other relevant information. So all the data are surface canopy. However, beginning in 2008, some of the data also includes subsurface kelp as a separate classification scheme. Next slide. So I'll briefly go over some of the CDFW uses of the data set, and these include during the marine protected area planning process to establish kelp habitat guidelines. It's incorporated into the Cal Coffee Stats to California current data set. And in 1989, the data was incorporated into CDFW's regulations for the commercial harvest of kelp. And the canopy area is incorporated into the kelp bed lease descriptions and calculations for kelp bed lease annual advance payments. Data is used to review proposed harvest during kelp bed lease requests and harvest plan review. And as you're aware from Cynthia's work, the CDFW Invertebrate Project also uses the data set with their work with abalone urchins. Next slide. So because the data set is because the data set is readily available online, it doesn't require any type of information to download. CDFW doesn't know everyone who uses the data set, only those who reach out to us for more information. So I've listed a few of the outside entities who have requested to use or requested additional information on the data set. And these include, universities, other institutions, and it's also been incorporated into the SynCoast data portal. Next slide. So this graph depicts the CDFW's aerial kelp surveys of giant and bull kelp from Marin County north to Del Norte County. So the vertical axis is the kelp canopy in the square kilometers, and horizontal axis is the year. You'll notice that this graph does not include all the survey years I spoke about earlier. So years which did not have a complete survey in the North Coast were omitted from this graph. Specifically, the 2009 and 2010 surveys did not provide coverage for the entire North Coast. And the North Coast was not surveyed at all in 2011 and 12. And 2013 did not have a complete survey. So as you can see from the data that we do have, the 2008 was a very good kelp year along the North Coast, which contrasts the low kelp year depicted from 2014. Next slide. So this graph depicts the CDFW's aerial kelp surveys in Sonoma County, and you'll notice that this graph includes more years of data than the previous. So based off of the CDFW surveys, 1989, 2002, 2008, and 2009 were good kelp growth years in Sonoma County. Sonoma County was not surveyed in 2011 and 12, and the 2013 survey did not include all of Sonoma County. So when the next survey was performed in 2014, you can see the drop in kelp canopy coverage, and 2015 had the historic low kelp year for Sonoma County. Next slide. So this graph is Mendocino County. For the years of data in this graph, 1989 and 2008 were good kelp growth years. Partial surveys were conducted in Mendocino County for 2009 and 2010, and there were no surveys in Mendocino County in 2011 through 2013. When the surveys were conducted in 2014 through 16, they depicted the low canopy that you see here. Next slide. So now I'll go over some of the value and the limitations of the survey. So value of the surveys include that these are a detailed 2-meter spatial resolution that allows for the depiction of giant kelp, smaller footprint of bull kelp, as well as small patches of kelp. So also it's a long-term data set proven to be very versatile and it's been used by multiple projects both within and outside of CDFW, and it is applied coastwide. Some of the limitations of the survey are that it does not provide estimates of kelp biomass, and because the surveys are annual, they're a snapshot in time, so they do not document the seasonal fluctuations of kelp. And as I mentioned earlier in my presentation, CDFW has not performed kelp surveys in some years due to lack of funding, technology, or staff capacity. And there are very few companies that can perform this type of work, and the costs have increased over time. So currently, CDFW does not have a kelp survey scheduled for this year. In 2017, CDFW did release a request for proposals. However, no bids were received. And the CDFW continues to discuss alternative technology, such as drones, satellite imagery, to capture this important data set. Next slide. So in closing, I provided an overview or some information on the CDFW's aerial kelp surveys, including brief background, how to access the data set, examples of uses of the data set, and a brief overview of the North Coast kelp surveys, and along with some value limitations of the survey. Next slide. And thank you. I'm available for any questions.
Excellent. Thank you, Rebecca. So if you have questions for Rebecca, go ahead and type them into that questions box in your webinar software. And so I have a couple of questions here for you from Steve Lonhart. His first question, Rebecca, is what error is associated with the canopy cover estimates?
Well, we don't have that kind of information. I mean, all of the data set are visual. Well, I actually don't have that information in front of me. I'd have to go back through and look, but you know, they're run through a specialized classification process, and then once they come out of that, there's also a visual that we do as well to try to pick up if there's any discrepancies, and we have found discrepancies in that. So I don't have that error right in front of me, but I can look into that.
Okay, thank you. And then Steve's second question is, what is the status of the drone-derived data?
I'm sorry, say again?
What is the status of the drone-derived data?
So we're currently not using drones to perform this kind of survey. We've been discussing internally, because of the increased costs with this type of survey, if we want to look at, satellite imagery, look at drones, but currently the department is not using drones to capture kelp canopy data. However, it's something we are discussing because we do have a drone program within the department. So that may be a method to use in the future for some of the areas.
Okay, great. Our next question is from Sarah Hamilton. You mentioned kelp bed leasing. Do people harvest Nereo in California?
Yes, people do harvest bull kelp in California. It's for human food. So in Northern California, all the kelp beds, administrative kelp beds in Northern California are closed. There's a few that are lease only, which means they have to apply through the commission. The commission, Fishing and Commission grants that. But there is in the North Coast some bull kelp harvest for human food. So the majority of the harvest now in California is giant kelp. So that would be Monterey South. And that would be for abalone food, for aquaculture farms.
Okay, thank you. And then we actually had one more question from Steve Lonhart. And that question is, are surveys done at about the same time of year?
Yes, so the surveys are typically done between August through October. There's been some years where they've been done a little bit sooner than that. We've even had some surveys go into early November. When we are able to do the surveys, we strive for a coast-wide survey. So if there were times when we weren't able to have a survey in a particular area, if we can get it in, and even if it's early November, we never went into late November, we would try to get that in.
Okay. Perfect, thank you. And then I have one last question before we move on to our next speaker. Okay, so this question is from Cindy Dawson from the Ocean Protection Council. When you say historic low, is that just for the years surveyed? And she says the early 80s have similar lows in some areas in at least some data sets like Jim Watanabe, sorry, I might be saying that wrong, in Monterey Bay. So is it a historic low just for the year surveyed?
Yes, yeah, thank you for that. I should have been more clear. I was talking about historic low when looking at the department data surveys that I had presented on. So yes, if we did not do a survey, then I was just discussing those particular data set for those years. And I was only discussing North Coast, or I was being specific as far as the different counties in the North Coast. So I was not discussing anything south of, San Francisco.
Okay, great. Thank you for clarifying. Excellent. Well, thank you so much, Rebecca. If anyone has any other questions, you can actually type them in the box, or you can save them for our discussion later. We're going to go ahead and move on to our next speaker, who is Tom Bell. So Tom, we're going to pull up your presentation and I'm going to unmute you here. Okay.
Okay, great. Thank you very much. So today I'm going to be talking about some of the research we've been doing at UCSB. I'm technically a postdoc right now at University of Alaska and UCLA, but I'll be transitioning back to UCSB in August. So I'm going to be going over a lot today, so please don't hesitate to ask questions at the end, or you can contact me directly after the webinar and we can talk about this offline. Next slide. So here's an outline of what I'll be talking about today. I'll be going over the methodology of estimating giant kelp canopy density and biomass along the coast of California. I'll be talking about briefly some new work extending these methods to bull kelp canopies in Oregon with Sarah Hamilton. And I'll be talking about some ongoing work using UAVs or drones to detect kelp canopy, both giant kelp and Nereus cystis. Next slide. And so giant kelp is a globally distributed marine macroalga and is considered a foundation species in that it supports an ecologically and economically important ecosystem. And today, again, I'll be focusing mostly off the coast of California. Next slide. And we have the ability to estimate canopy density and biomass of this species from the Landsat satellites. And so we can do this starting with Landsat 5 thematic mapper, which was launched in 1984 and flew till 2011. And we have a continuous time series over the past 35 years, giving us the ability to look at kelp canopy dynamics back through time. And when we have two sensors flying, like we do right now with Landsat 7 and Landsat 8, we get an image every 8 days, regardless of cloud cover, and a usable image of most areas in the California coastline every month. And these images allow us to look at kelp canopy on a 30 meter by 30 meter pixel scale. And we could actually look at fractional cover inside these 30 by 30 meter pixels, which I'll discuss in a minute. Next slide. And so in order to ground truth our biomass estimates, we rely on a time series of canopy biomass started by the Santa Barbara Coastal Long-Term Ecological Research Project down in the Santa Barbara Channel. And they started back in 2002. And so here researchers have developed an allometric relationship between the number and lengths of different frond segments to estimate the biomass of giant kelp in several 40 by 40 meter plots in the Santa Barbara Channel. Next slide. And so for this talk, I'm going to focus on the area between the US-Mexico border and just north of Santa Cruz, California, which is shown by the white dashed lines in this image. And this includes the offshore Channel Islands and represents the region where giant kelp is the dominant canopy-forming macroalga in California. And this is about 1,500 kilometers of coastline, which represents 8 Landsat tiles. Next slide. And so here are a time series of kelp biomass at 3 sites as estimated by Landsat. And so the top time series shows the extreme seasonality of kelp canopy that is typical of the Monterey Peninsula and Central Coast, while the bottom time series shows the interannual patterns that are typical in areas around Orange County and San Diego. And the middle time series shows one of our validation sites, the MO or Mohawk site. The blue represents the LTER diver surveys, and the red represents our time series. Next slide. And so in the past, we've manually classified kelp canopy using false color, near-infrared false color images, which we see in figures A and B here, and this is similar to what we saw in the last talk. And so these red areas show regions of high near-infrared reflectance, which were then manually identified by mostly undergraduate analysts. And so the canopy and once these areas were identified, the canopy density in these areas were modeled using multiple end-member spectral mixture analysis, or MESMA. And we model each 30 by 30 meter pixel as a linear combination of a pure kelp spectral end member and 30 seawater end members. And this gives us an estimate of the fractional cover of kelp canopy in each pixel. We can see fractional cover using Landsat down to about 10% cover of that 900 square meter pixel. And I won't be going into the details of MESMA today, but feel free to ask me about it. Next slide. But while the Landsat sensors represent an uninterrupted time series, there are important differences between them. And these include increases in the signal-to-noise ratio as we move through time, and the quantization of data. So for example, Landsat 5 and Landsat 7 sensors were 8-bit. So we see reflectance in 256 different digital numbers, but Landsat 8 is a 12-bit sensor. Next slide. And there's also important spectral differences between the sensors. So for example, the top line here shows the Landsat 8 operational land imager, or OLI. You can see the near infrared band, which is that pink band labeled 5, and it's significantly thinner than the near infrared bands in Landsat 5 or Landsat 7, which are the 2 lines below it, where the near infrared band is labeled 4. And they did that to avoid this absorption feature from water vapor. Next slide. And so to correct for these differences in spectral bands between the sensors, we simulated Landsat 5, Landsat 7, and Landsat 8 imagery from a hyperspectral image that was taken of kelp canopy in the Santa Barbara Channel in 2013. And this allowed us to generate kelp fraction estimates as if the kelp forest was measured by the 3 Landsat sensors simultaneously. And we see there was an underestimation of kelp canopy fraction by Landsat 8 compared to Landsat 5 and 7. And we were able to correct for that. So if you look at the bottom row, the OLI_C, that's our corrected Landsat 7, or sorry, corrected Landsat 8. So Landsat 8 kelp fractions line up to the 1-to-1 line with Landsat 5 and Landsat 7. Next slide. And so we then compared our kelp fraction estimates from MESMA to canopy biomass estimated from divers at our validation sites for each sensor and for all sensors together. And we transformed our kelp fraction estimates to kelp canopy biomass from Landsat. And next slide. And so we have a manuscript in revision at Remote Sensitive Environment, which describes how we recently automated this protocol and took away the manual processing step. And so this process allows us to transform a Landsat image into estimates of kelp canopy biomass in just about a minute, which used to take a human about an hour to do. Next slide. And so basically, I'll give you a quick rundown of how the automated process works. We start with a level 2 reflectance product from USGS. We stack 6 different bands across the color and near infrared and shortwave infrared regions of the spectrum. We then mask out land using a digital elevation model from the Aster satellite sensor. And then we use binary decision trees to classify each pixel as seawater, cloud, or kelp canopy. So you can see the kelp canopy off Santa Rosa Island here in yellow. And so this step removes the need for any manual classification. And we then use MESMA to estimate the fractional cover of kelp in each pixel identified as kelp canopy by the decision tree process. And we apply our empirical relationship with the diver data to transform those pixel fractions to canopy biomass. So you can see the differences in canopy biomass density in step 3 there. Next slide. And so these are confusion matrices between our manual and automated classification processes. And you can see that the process does quite well for each sensor. TM is Landsat 5, ETM plus is Landsat 7, and OLI is Landsat 8. And then when we add all the sensors together, we still do pretty well. Next slide. And here we're comparing the time series of kelp biomass in the Santa Barbara Channel from our manual classification in blue and our automated process in red in figure A. We can see they match up fairly well. In figure B, it's a scatter plot between those 2 time series. And there's a slight underestimation by our automated protocol versus our manual. And we think this is because when we went back through the manual images, we see that sometimes the analysts had identified intertidal surfgrass or intertidal algae or waves as kelp, which were not identified as kelp by the automated process. So we're fairly confident the automated process actually does a better job than the manual process does. Next slide. And so one final part of the process is the correction of the Scanline Correction error lines from Landsat 7 imagery. And so these were caused by a mechanical failure of the sensor, which leave black missing data lines across the images. So here we see San Miguel Island in figure A and Point Sur in figure B. And you can see those Scanline corrector error lines going across them. And these vary in width as you move from the center of the image out to the edge. And they also move in space from image to image, which we actually use to our advantage. Next slide. And so we've taken advantage of the spatial synchrony of kelp canopy to help us fill these lines. And so what I mean by spatial synchrony is that giant kelp, if you look at giant kelp pixels or giant kelp beds, kelp canopy is going to vary through time more similarly in beds which are closer together than farther apart. And since the scanline error lines move from image to image, we can fill mixing pixels using known biomass relationships between the missing pixel and nearby pixels, which are not covered by the missing data lines. And so what we did here is we took Landsat 5 imagery, we put missing data lines over them, and then we filled the gaps, and then we compared them to the actual images. And next slide. And so the synchrony gap filling validation shown here in red in figure A does fairly well on the pixel scale. And it does quite well when pixels are aggregated to a patch scale, which we see in figure B. Okay, next slide. And so finally, I want to turn everyone's attention to the effect of tide on kelp canopy estimates. And this is going to be a problem when you use any satellite image or aerial image. And so what we did here is we compared Landsat 5 to Landsat 7 biomass estimates from 3 Landsat tiles in Southern California from 1999 to 2003. So this is before the scanline corrector failure. And so Landsat 5 and Landsat 7 images are taken 8 days apart. And we found that in some Landsat tiles, Landsat 7 showed greater biomass than Landsat 5, and in other tiles, the opposite was true. And so this was kind of perplexing because we first thought it might be a difference between the two sensors. And so what we found was that due to tidal cycles linking up with the 8-day offset between the sensors, which differed by region, that kind of controlled whether or not Landsat 7 was seeing more than Landsat 5. And so we corrected for this. We can correct for it, which is shown in figure C. What we ended up doing is just looking at a seasonal average for each site. Next slide. And so we've used this automated protocol to develop a seasonal time series of every giant kelp pixel in the range of dominance in California. Next slide. And this updated data set will be available through the Santa Barbara Coastal Long-Term Ecological Research Project website with the publication of this new paper in the next month or two. Next slide. Okay, and so I wanted to talk a little bit about some ongoing work I'm doing with Sarah Hamilton up in Oregon, where we took a look at the Oregon Department of Fish and Wildlife aerial photography and compared it to the Landsat estimated kelp canopy fraction for Nereus cystis. And we see that there is a fairly strong relationship between the two. So we're moving ahead with making time series in Oregon with the automated protocol. And we'd like to do the same for Northern California as well. And I'm working on a time series for Southeast Alaska right now. Next slide. And so because we have this Landsat continuity, we can kind of look back through time, back to 1984 at any site we want in the United States and most sites around the world. And next slide. And so finally, I want to talk a little bit about how we've been using drones to image kelp canopy. So these are some, I flew about 100 drone flights in Alaska last summer, where we have both Macrocystis giant kelp and Nereus bull kelp. And you can see the drone images on the left and our classification algorithm to look at emergent canopy on the right. And you can see the differences between giant kelp and bull kelp canopy. So I've been doing a lot of drone work in Alaska and Santa Barbara, and I think there's a lot of potential for this moving forward. And next slide. And I'll be happy to take any questions, and feel free to email me or go to my website. Thank you very much.
All right, excellent. Thank you so much, Tom. So we do have a question from Tom Ford. Are there restrictions to crosswalking this for bull kelp? And you mentioned Oregon. How do you account for depth?
Well, we're only looking at the canopy.
Okay. And for the restrictions to looking at bull kelp?
Restrictions, what do you mean by restrictions?
So
Like,
the specific question, and Tom, Tom Ford, you might need to clarify. He says, are there restrictions to crosswalking this for bull kelp? And he said, you mentioned Oregon.
crosswalking this, I'm not sure. There's limitations for kelp, for bull kelp for sure. It presents less of a surface canopy than macrocystis. It's probably also could be greater subject to tide and current.
Okay. And Tom Ford, if you want further clarification, you can type that in there. Oh, he said meaning biomass as it's related to depth, and yes, the limitation is
okay. Yeah, so there's not a lot of biomass surveys done for bull kelp. If anyone knows of any long-term biomass surveys, please let me know so we can validate this. Right now, we're just kind of looking at canopy density because our validation we're using are the higher spatial resolution overflights that were done by the Oregon Department of Fish and Wildlife.
Okay, great. And there's a question from Mark Carr. Why doesn't aerial versus Landsat relationship for bull kelp in Oregon go through origin? Tom, do you want me to repeat the question? Tom?
Oh yeah, go ahead. Sorry, I lost you for a second.
Oh, sorry about that. The question from Mark Carr was, why doesn't aerial versus Landsat relationship for bull kelp in Oregon go through origin?
So let me pull that up on mine again, just so I know. So it doesn't, and it doesn't. We think that's because there is at very low canopy densities. There's a lot of error on whether or not we can detect bull kelp at all. So when we're getting to maybe 5% of the pixel covered with bull kelp, we probably have trouble observing it at all. We would also want to test our kelp end member. Here we just used a general brown kelp end member. Spectrally, bull kelp and macrocystis look almost exactly the same. But we'd probably want to try to look at a bull kelp end member taken from either Landsat imagery or from the lab and see if that does any better.
Okay. Next, I have a question from Helen Berry. Yep. She says, thank you for your talk. Have you estimated the threshold of bed sizes that the Landsat imagery captures?
Yeah, so we think, for giant kelp, we can go down to about 10% of the pixel. So as long as there's about 90 square meters of emerging kelp canopy inside a 900 square meter Landsat pixel, we should be able to detect it. And I imagine that would be about the same for bull kelp as well.
Okay, great, thank you. And then I have one last question from Rebecca Flores Miller, and she says, could you discuss the costs of obtaining the satellite imagery?
The satellite imagery is free. And with the automation protocol, it takes us a lot less time and effort to do it. Yeah, we'd be happy to work with the state and make these data more publicly available.
Okay, excellent, thank you. So again, thank you, Tom, so much for your talk. If anyone else has any other questions, feel free to type them in there or save them for the discussion. We are going to move on to Meredith and her talk. So I'm going to go ahead and unmute you, Meredith. Give us just 1 second. All right, perfect. So Meredith, take it away.
Hi everyone. Good morning. Thanks for inviting me to present. So today I'll be discussing some of my research that actually builds on a lot of what, not builds, adds to, I guess, what Tom just talked about, and methods that he and Kyle Kavanaugh have published, but applied to bull kelp, and of course, mostly looking at Mendocino and Sonoma counties. Next slide. So when we think about remote sensing of kelp using satellites, giant kelp pops into our minds usually, and that's because remote sensing studies of bull kelp remain largely absent from the published literature. And because of this, there are several important considerations to take into account between bull kelp and giant kelp. The first, which is the life cycle history. So bull kelp is an annual plant and giant kelp is a perennial plant. So we only see a canopy for bull kelp during the months of August and October or November. And where giant kelp has a seasonal maximum in the summer and early fall, we can detect it year round with satellites, where we're not necessarily able to do that with bull kelp. There are also important morphological differences between the biomass distribution for bull kelp and giant kelp. Bull kelp has biomass concentrated at the surface and the canopy, where biomass is distributed, or giant kelp biomass is distributed throughout the entire water column. And I think for me, one of the most interesting science questions is what the response to climate change is between the two different species. Next slide. So there's a paper published by Dan Reed and others that challenged the thought that giant kelp is a sentinel species of climate change. And so we can see on the figure on the right, where we have biomass on the y-axis and annual temperature anomaly on the x-axis from -1 degree Celsius to 2 degrees Celsius. There wasn't really an impact of temperature anomaly on giant kelp biomass, but they did see significant impacts to sessile invertebrates like sea urchins and sea stars. But of course, we know that there's been a dramatic response by bull kelp in Northern California to some combination of environmental drivers over the last 10 years, but whether or not that was just a response to temperature is yet to be understood. So I think that remote sensing is an excellent tool for us to be able to parse out these environmental drivers that have impacted kelp beds in the Northern California coastline. Next slide. But of course, the theme of our discussion today is remote sensing and using it as a tool to understand and monitor bull kelp. So one of the primary focuses of my research is to understand whether the impacts of spatial resolution impact our ability to measure bull kelp after the die-off. So in this cartoon here, we see a 30 by 30 meter pixel, which would depict Landsat. And it has proved to be a fantastic tool, as Tom has shown us already, for detecting relatively large kelp beds, mostly giant kelp and what Tom showed for Oregon. But we see that bull kelp, after the die-off, the diameters of the bed were reduced to very small, in some cases, single plants, less than 10 meter patch sizes. So it's unclear whether or not Landsat is a tool that can be used to detect these small patches. Next slide. So I'm looking at satellites with higher spatial resolution, like the example on the right, which has a 5 by 5 meter pixel resolution. And you can see that it increases the likelihood of small patches of bull kelp being detected. Next slide. So as Tom already talked in great detail about and gave great examples, Landsat is a good historical record. And the 30-meter resolution is adequate to measure giant kelp, but is it adequate to measure bull kelp after the die-off? And so there are a bunch of sensors coming online, and even some that are not really necessarily young, that have higher spatial resolution and could be useful to monitor smaller patches of kelp. So if we look at the figure on the right, we're going to ignore the middle panel, the pan sharpened, but the top panel shows a 2-meter resolution sensor, and the bottom master shows a 50-meter resolution. There's almost a 2-fold difference in the estimation of giant kelp canopy in Santa Barbara between the two sensors. So one of the significant challenges is how spatial resolution will influence our ability to measure bull kelp. And increasing our spatial resolution is important because that means we're accurately measuring biomass and the impacts of these environmental perturbations on kelp canopy. Next slide. Looks like we had a little bit of a mess up with the figure, but that's okay. So I conducted a sensor comparison between RapidEye, which is a commercial satellite, Landsat, and the CDFW aerial surveys. And so I'm looking at our CDFW admin bed number 307, which is basically Van Dam Bay to 10 Mile Estuary. It's a small portion of the coastline because I'm still just testing out this new imagery. Landsat has a 30-meter resolution pixel size, and RapidEye has a 5-meter resolution pixel. So I analyzed for kelp canopy coverage using methods developed by Roberts et al. Tom had discussed that in his talk, called MESMA, and has been applied to kelp by Tom and Kyle in multiple publications. So these are preliminary results from CDF, from a comparison between these 3 sensors, and the figure on the right showed that the 3 sensors are spectrally similar, but RapidEye does have 1 additional band in the red edge, which is this really steep portion of the brown kelp spectra that Landsat does not have. Next slide. So just to go into a little bit more detail on the RapidEye satellite, which I'm sure most of you have probably not heard of or used. It's a commercial, so it's run by a commercial company called Planet Labs in San Francisco, and it has a relatively good record from 2009 onward. And I'm a part of the research and education program, which provides 10,000 square kilometers of free imagery per month, which is a lot and probably more than I will ever use, but it's a great program to be a part of, and they provide a lot of support to the people who are members. And as I already mentioned, it has a 5-meter spatial resolution. In comparison, Landsat is non-commercial, run by USGS and NASA, and has a continuous record from 1984 onward, and is free, unlimited to anyone with an account. Next slide. So here I'm just showing some preliminary results for the sensor comparison between RapidEye, Landsat, and CDFW. So CDFW is in blue, Landsat in green, and RapidEye in red. And we have kelp canopy, for the figure on the right, we have kelp canopy coverage on the Y and the year that we have overlapping data on the X. So as you can see, Landsat and CDFW match up pretty well, but RapidEye doesn't, and overestimates for the years that we have overlapping in 2015 and 16. And for the figure on the right, where we have satellite canopy coverage on the Y and CDFW canopy area on the X, we get a pretty strong relationship between Landsat and CDFW, but it's probably being driven by the point in the upper right-hand corner. And of course, as I already mentioned, rapid eye appears to be well outside of that relationship. So I don't really want to discount the results that we have from rapid eye yet. It's a new satellite that we're still learning to work with. Next slide. So as I mentioned, these are preliminary results. We're still learning to work with the rapid eye imagery. There are several challenges that I've encountered in addition to just being a student and learning how to work with satellite imagery, where there's no atmospherically corrected product for rapid eye imagery. So we're conducting all of the atmospheric correction methods ourselves, and signal-to-noise ratio is important. Usually for higher spatial resolution satellites, the signal-to-noise ratio decreases, which may impact our ability to measure kelp or anything with these high-resolution commercial satellites. In addition, it's much more computationally expensive to work with the rapid eye imagery than Landsat. For every Landsat pixel, there are 36 rapid eye pixels. So you can imagine how much that impacts the time to process imagery. So as I move forward, I'm refining, this method of detection of kelp with rapid eye imagery. And ultimately, I hope to include other ultimately I will include other satellites, some commercial, some not, into the sensor comparison to help us understand what the impact of spatial resolution of the satellite is on our ability to detect it. Next slide. So now I'm going to transition into some of the biomass estimations that I'll be making this summer. Next slide. So from these photos, you can see that on the left is a giant kelp canopy, and on the right is a bull kelp canopy. And that they look distinctly different, and that impacts what we see from space. And also, as Tom discussed in his talk, tide implications are important between the two. So because the biomass distribution of giant kelp is more even along the stipe than it is for bull kelp, there's probably less of an impact of tidal fluctuations on what the satellite sees. And as you can see in the bull kelp picture, there's only a single gas bladder, and there are no pneumatocysts that create this canopy of blades on the surface. So for each pixel, the fraction of water to kelp is much different for bull kelp than it is for giant kelp, and an important consideration to take into account when we're doing remote sensing of bull kelp, and also when we're making our biomass measurements. Next slide. So on a first order, as we already talked about, there are not really any biomass estimations for bull kelp really anywhere, other than one paper published by Spectral in Alaska. However, we're working in California, and so I'd like to develop a biomass estimation for California bull kelp, since we're not certain that the relationship developed by Spectral et al. Would hold true for California. So Spectral et al. Measured several morphological characteristics of bull kelp including the longest blade length, the bulb diameter, and the subbulb diameter, and found that the subbulb diameter was the best predictor of total plant weight. So basically they collected hundreds of plants and weighed them throughout the growing season. But my plan is to pull these plants out at the end of the growing season when the winter storms would rip them out anyway, and then develop the biomass estimation based on these plant morphology characteristics and plant weight once we know that the plants are going to get ripped out anyway. And these plots in which I'll conduct the measurements will correspond with a Landsat pixel, a 30 by 30 meter plot, so that we can correlate biomass estimates from the in situ measurements to our satellite. And next slide. And these types of relationships between biomass and optical properties or satellite products has already been developed by Stiegel and Kyle Kavanaugh. So using either the fraction of kelp per pixel and the transport canopy biomass for giant kelp, or a band ratio for bull kelp. And because I'm using MESMA, multiple in-member spectral mixture analysis, I'll most likely develop a relationship between kelp fraction and biomass. Next slide. So in addition to that, I'll be building on work that Tom Bell did in measuring kelp health by taking in situ measurements of chlorophyll and carbon and developing a relationship in which he could detect that from a remote sensing platform. And I am actually hoping to optimize this for a multispectral sensor and to put that on a drone. And hopefully that will give us flexibility to fly the drone wherever we want, do long-term studies of different regions or bays, and monitor kelp health at very high spatial resolution. Next slide. So in conclusion, I'm really hopeful that we can learn as much about bull kelp systems as we have about giant kelp systems in Southern California, using a lot of the techniques that Tom is currently working with. And that I think that we should not put all of our eggs in the Landsat bucket yet, that we should consider these higher resolution commercial satellites, and that companies like Planet Labs are revolutionizing scientists' ability to get access to high resolution imagery, which is great, and I think more companies should do it. And that drones can help us monitor these areas, especially very cloudy areas like the northern coast, and help us get a greater physiological understanding of these systems. Next slide. And thank you, and I'll take any questions.
Excellent, thank you, Meredith. So if you have questions for Meredith, go ahead and type them in that question box. We do have one from Steve Lonhart. He asks if there is information available about latitudinal variation in aerial biomass, and for some context, he mentions that he's noticed that Nereocissus up north and into Washington is much less robust than along the Big Sur coastline. So is there latitudinal variation or information about it?
So the only study that I know of regarding biomass of bull kelp is the one that I mentioned in Alaska. I don't think that it's common for people to take these measurements. And so I don't know of any study that is published that shows a trend in biomass along the coastline, but maybe Mark knows or something.
Okay, great, thank you. So we are actually going to take one more question here from Helen Berry. So she says that she's excited to hear that you're looking at the Nerio biomass estimation. She thinks that Steckel found that biomass ground truthing is very expensive and that there was significant variation by location. How do you plan to address that? Lots of different locations.
Yes, so I actually forgot to mention that I am doing all of this work this summer with Cynthia and Laura up in Mendocino and Sonoma counties, and we are visiting several sites along the coast in which that will allow me to get this pretty large spatial variation of biomass across the two counties and across the CDFW sites. And I can correlate that to specific, because I'll know the locations of the Landsat pixels and I'll know the locations in which I'm taking my measurements. I can correlate those directly back to the satellite. Does that answer the question?
Yes. No, excellent. I think you addressed the question. So, and then Cynthia just mentioned that she thinks that Katie Dobkowski, sorry, I'm massacring people's names today, at the University of Washington did some morphometry measurements as well. Excellent. Okay, perfect. So thank you for the questions, and thank you, Meredith, for your talk. That was a lot of really excellent information. And we are actually going to take a break for the next 10 minutes. We're about 3 minutes past our planned schedule, but I think we'll go ahead and take the full 10 minutes so folks can have a bit of a break. And then we'll come back together at 11:23 for Mycera's talk. So we'll go ahead and mute all attendees at this time and talk to you all in just a few minutes. All right, excellent. Welcome back, everyone. Hope you enjoyed our break. So our next speaker is Mysira Costa, and if she has her with her, also Sarah Schroeder. They're going to be talking about the detection of bull kelp in the Salish Sea using high-resolution satellite imagery. So Mysira, I'm going to go ahead and unmute you right now.
All right, thank you, Rieta. Can everybody hear me? Hello, can you hear me?
Yes, your audio is perfect.
All right, thank you. So as Rieta mentioned, this is part of the work that Sarah is doing with us in my research lab at the University of Victoria. And yeah, I'll show you, it builds up on the previous presentations because I will show you some of the work that we're doing in the coast of British Columbia, mostly related to use of satellites for detection of bull kelp. Next slide, please. First, I'll just give you a little feedback on the context of the research that we're doing, which includes the kelp part as well. So this is part of a large collaborative project, which includes colleagues actually from the US, from Washington, and also colleagues from British Columbia, looking at the decline of Chinook and coho salmon, also at sockeye salmon as well. And as part of this collaborative research, we are focusing on the productivity of the ocean, the coastal ocean, on eelgrass, spatial and temporal variability of eelgrass along our coast, and the kelp part. So I'll be focusing on the kelp part for this talk with you. Next slide, please. So I'll give you just a little snapshot of the other components of this large project, which most of the data that we use come from satellites, and I name a few of them there, and I highlight in red the one that we use for the kelp component of this project. We have sensors aboard of the British Columbia ferries that cross the Salish Sea daily. We have also data collected aboard of research vessels, and we also have data collected by citizen science aboard of the BC ferries as well. And again, I'll be focusing on the little red circle, which is on the top right of this slide. Next slide, please. So for the ocean productivity, as I mentioned, we have sensors aboard of the BC ferries, and we have this really cool sensor that continuously detect light coming from water, reflectance, similar measured by the satellites. And we use this data for validation of atmospheric correction of oceanographic satellites, such as Sentinel-3, VIRS, and MODIS Aqua. Next slide, please. And from this kind of proper calibration of the atmospheric effects on the image, we end up having these very amazing, beautiful maps of chlorophyll concentration in the waters of British Columbia and Alaska. And we're using this data to define biogeochemical domains along the migration route of juvenile salmon. And this is data that will be associated with zooplankton diversity and abundance along our coast and the health of juvenile salmon. Next slide, please. On the eelgrass part, this is a work that we have just kind of finalized a major component of it with Natasha Narnik's work. And she looked at aerophotography and UAV data for most recent to define spatial temporal trends on distribution of eelgrass along the coast. And here is just an example of one of the regions that she looked at on the Gulf Islands from 1932 to most recently 2016, in which the 2016 data was derived by UAV, and the green area is the area mapped as eelgrass. And the other data are all from aerial photography, archived aerial photography from the region. And then from this type of information, she also associated declines with shoreline development that were also derived from the aerial photography. So it's quite a beautiful piece of work and very unique. We didn't have this kind of work in this region. So it's quite interesting. She just finalized some papers about that. Next slide, please. So in the kelp front, Sarah, who is conducting most of the research right now in this field, is looking at presence of juvenile salmon on kelp beds, bulk kelp, along our coast here. So last year she did a lot of snorkeling and underwater video survey on kelp beds, and this data is being analyzed right now, showing some very interesting results. Next one. And the other component is what I'm going to be focusing on this talk, is about detection of bulk kelp with satellites. So for this component, what I would like to highlight, which both Tom and Merritt mentioned it already, there are several variables that play a role on the ability of using satellites to detect kelp. And I kind of summarize some of the most important ones for bulk kelp. So the kelp structure is important, and for that, we would like to highlight density and exposure, and I'll go through this in little detail with you in a minute. The environmental characteristics of the area, such as tide height, which was already mentioned, time of the year, and the properties of the water surface, and I'll show you what I mean by that. And also the satellite characteristics in terms of spatial and spectral resolution, which both Tom and Merritt already mentioned as well. Next slide, please. So in terms of the kelp structure, we have a little schematic diagram here. And again, keep in mind, this is not to scale. What could be a cross-section of bulk kelp? And so you may have this 10 meters long kelp, and the reality is what we'll see on the surface is what is above the surface line there. It's mostly the bulb and the part just below the bulb. So there's not too much information or not too much structure of the kelp that is exposed and possibly detected by the satellite. The satellite will detect a little bit what's under the water, but definitely not as much as what is above the water. So you end up having the two signals sometimes mixed, what's above the water and what is slightly underwater, and also represents kelp. So the next slide, please. So this is what possibly the satellite would be seeing. So this is the aerial view, like if it's a bird flying over the area. So the emerged part is what is in white, and then you have all the more dense parts of the kelp are all submerged. And that is one of the limitations of using satellites for detection of bulk kelp, is the fact that not all the components of the kelp structure are exposed or they are above water. Next slide, please. So the tide height, which is related to the environmental characteristics, together with the kelp structure itself, will play a role on the detection ability of the satellite. So what you see in your left side there is the ideal situation is when your image is acquired when your tide is low. Right, so you can have some of the blades and some of the structure of the kelp exposed, and that's the ideal situation. So if your image is acquired during a slightly higher tide, which is your figure to the right, then you don't have any kelp at all. So your kelp map would say that, okay, kelp disappeared that specific year, and this is just related to the time in which the satellite acquired the image and the tide height at that specific time. So anytime that you have to download historical data or plan an acquisition of a satellite image, one of the main important components is look at your tides, because that will guide you if you will produce a good or a bad map of kelp. Next slide, please. The time of the year becomes quite relevant, especially for bulk kelp, because it's an annual species, as Merritt had mentioned before. So if you acquire an image like on the right side figure, you will see it's too early in the season and there's no kelp in the region. right? So it doesn't mean it's just not exposed at that specific time of the year. And in our situation here, for example, if you're acquiring in July, August, then you have maximum growth and extent of kelp. So if you combine images acquired at this time of the year with the low tide, then you have the ideal situation for proper detection of kelp with the satellite image. One of the things that I would like to point out here is also for you to pay attention on the proximity to the coastline. And that becomes a problem when we think about the pixel resolution of the satellite and what we normally use as a jargon called spectral mixing. So when the spatial resolution of the satellite is slightly higher, and for our situation here, where the bulk kelp happens, I would say, for example, the Landsat 30-meter spatial resolution, what you have along the coast is a pixel mixing. So you have rocks or vegetation plus water plus kelp in a 30-meter spatial resolution. And it's not that easy to detangle the spectral signal from all those different targets along the coastline. So the spatial resolution, at least for the BC coast, is very important because the bulk kelp happens very close to the coastline, and a large pixel adds a lot of error on identification of kelp, because those pixels contain information, reflectance information that are related to the coast itself, to the land, to the vegetation or the rock or sand beaches, whatever it is in those regions. Next slide, please. And the other environmental characteristic that it's important, but that's a function on, it's a function of how the time of the year or the time of the day and the position of the sun when the satellite image is acquired. So we call that glint. And basically that's what you see in this example in the image here. This whitish on the water, it's all glint, which is kind of this meter effect when the satellite viewing angle is similar to the sun angle, just opposite directions. And it's very difficult to correct for that when it's happened, such as the example that I show here in this slide. And when this happens, most of the information on anything that has to do with the water or kelp is lost. So that's another component that we always have to be careful when we plan a satellite data acquisition or when we look at archive data, because they may be cloud free, but if glint prevails in the image, so it prevents you to use that specific image. Next slide, please. And lastly is to understand the characteristics of the satellite in regard to its spectral resolution and the kelp reflectance. So Sarah has done several in situ sampling using instruments similar to this one that you see in the boat there highlight, which are hyperspectral above water sensors. So basically they are small satellites, but they are in a hyperspectral domain instead of multispectral, such as most of the satellites that we use these days. So you have much higher resolution data, spectral resolution data acquired. So I'll show you in the next slide example of that. Next slide, please. So this is example of data that Sarah collected last year, actually, or this year, no, last year, yes, at different kelp beds looking at different densities. So what you see in the y-axis is reflectance, which is the light coming from the water in percentage to make it easier to understand. And on the x-axis is the wavelengths of light. So you go from blue, and I highlight with a blue color bar, green, red, and I put gray for the infrared spectrum because we can't really see that. And the different plots there, they represent different densities, in which the brown one is for very dense kelp, sparse dense is kind of the slightly brownish, and a pink for the submerged, and the reflectance for water is your blue curve. And what you notice is the water has very low reflectance, especially in the infrared part of the spectrum above 700 nanometers due to light absorption by the water molecules themselves. And kelp, regardless the density of it, shows higher reflectance in the infrared part of the spectrum. And if you have a very high density, such as the dark brown curve, you will see it will affect the end of the green and the beginning of the red spectrum due to the pigments on the kelp itself. What we have to think about is that the condition that the kelp has in our coast here is mostly sparse and submerged. It's very rare that we'll see dense kelp as showing in this picture here. So we have to figure it out how to use this infrared and red part of the spectrum to acquire or to map the kelp in our waters. Can you pass the next slide, please, Vieta? So I zoom in that area. You see here at the end of the red spectra and the near infrared spectra are the main parts of the light field that we can actually effectively use for detection of bulk kelp. Next slide, please. So knowing that, we select a few satellites with high spatial resolution. So some of the satellites that we're using, it's Worldview 3 and others satellites. So we build up a time series from 2004 up to 2016. And we are analyzing this data in a way that we can define what we call spectral fingerprint or spectral kelp print for kelp, and using this for classification. However, before this is actually accurately done, we have to process the data properly. So what I'm showing here to you in your left, it's like a little piece of Worldview 3 image at a 1.2 meter spatial resolution. And just as a curiosity, this data is acquired at an altitude of about 600 kilometers above the Earth's surface, which is the orbit of Worldview 3. So you see it in true colors, that's what we would see. And then you see on the right side, false color. That's when I add the infrared spectra on the red channel of the display. So you see the difference between the two, but not necessarily you can see kelp everywhere. So next slide, please. So this is the same area after I processed, or actually Sarah processed, and then you start to see the kelp, which is the red areas along the coast. So I'm going to try to go through the steps to reach this point of the analysis. Next slide, please. So first, the first thing that we have to do is correct any inaccuracies related to geometry, because we're using different satellites. So we may have geometric inaccuracies, and we want to make sure that all the images overlap. So that's kind of the first step. And after that, we have to correct for effects of the atmosphere and glint. Glint is that signal that I mentioned to you in previous slide, that it's very hard to correct, but depending on how strong it is in the image, if it's not that strong, you can still correct it. And because we're doing temporal analysis, we make sure that we do a spectral normalization of all our historical data. And after that, we do some processing to remove land and deep water to minimize possible interference of land and deep water on the classification of the kelp itself. Next slide, please. So this is just an example of the atmospheric correction. So what you see in the left there is the digital numbers in the y-axis, and again the spectra on the light wavelengths on the x-axis before atmospheric correction, and the plot represents Kelp. And on the right side, it's after atmospheric correction for the same target for Kelp. So what you see is a dramatic change in the spectra, and the differences are due to atmospheric attenuation. So after you correct for the atmosphere, you remove all the atmospheric attenuation, which means how the particles and gases in the atmosphere absorb and scatter light. So you remove that component and you recover the true spectra of Kelp, which is quite similar to the one that we measure in the field. Next, please. This is a little example of glint correction from one of the images. So again, what you have in the left, it's a non-glint corrected image, and on the right is your glint corrected image. So it's quite effective. It gets a little bit complicated sometimes in areas that have a lot of glint. So the lower plot represents a transect along that red line that shows on the top plots for the RGB bands of that specific image. And you can see the variability on the left plot is much higher than the variability on the right plot. And that variability on the left plot, especially the upper values, the higher value, the spike values, are due to glint. And then this is very effectively actually removed on the right side image. Next slide, please. So after you do all these pre-processing steps on your image, so you have your best image possible, the next step is working what we are calling the spectral Kelp print. So we work with different band ratios, contrast. We develop and look into different spectral indices, combining different bands of the satellite that highlight the Kelp signature. We work with principal component analysis as well. And then after that, Sarah did a strong statistical analysis using different statistical indices to define which are the best spectral Kelp print that characterize Kelp effectively. And I'm just listing some of this here: the GNDVI, the principal component 2, and the near infrared band by itself. Next slide, please. So this is a combination of those three previous spectral indices that I show in the previous slide. And what you see is those areas in pinkish and reddish are all areas that correspond to Kelp. Next slide, please. So from that, we did two classification approach because we're testing which ones would work better. And on an image from 2016, which we have a lot of in situ data collected with a kayak survey, we did a supervised and an unsupervised classification, so we can actually test the accuracy of the different methods. So in your left side, you see the supervised classification results in which the green polygon shows Kelp, and the unsupervised on the right side. I think I forgot to put it here, but the classification accuracy for the supervised classification was about 82%, and the unsupervised was about 80%. So it was very good accuracy for both. But there are some complications on that because slightly difference between when the kayak survey was conducted and the time in which the image was acquired may change the classification accuracy. So we have to be very careful how to evaluate that. We think that possibly the accuracy is even higher. It's just the slightly lower accuracy that we see, which I don't think it's lower because it's all above 80%, but this number is a reflection also of the mismatch between kayak time acquisition and satellite image acquisition. Next slide, please. Okay, so yeah, so here you go. Here's the kayak survey. So you have your unsupervised classification here on your left, and on your right, the green polygons represent the polygons derived from the kayak survey. So it's a very, very good match, and we have data for this for several regions. So we're going through the different regions right now to understand this better. And why we want to make sure the unsupervised classification gives us good results is because the historical data, we do not have in situ data to train the classifier and do a supervised classification. So we want to make sure that the unsupervised classification is robust enough, and then we will apply that to the historical data from this region. Next slide, please. And that's what Sarah did. So here are examples of some of the historical data. So the first one is 2004, and then 2012, 2015, and 2016. Sarah is working in other years for this time series for the different regions. And the red polygons are the one derived based on an unsupervised classification for this region. So how we intended to validate the historical data, because we do not have in situ data for all the regions that we're working with, is one, we're going to use what we call those spectral kelp print, and use that as a sign that we're accurately classifying kelp. So we're going to go in those areas, let's say in 2004, and find the same signature that was defined for 2016. The other source of data is by association. So those shorelines, are they possible to have kelp? If it's a beach, likely not. If it's rock, yes. And we also use data from another program called Shore Zone, in which they delineate areas that have occurrence of kelp along the BC coast. So we're trying to maximize the amount of ancillary information that can help us to validate the historical data. Next step, please.
Hey,
Next slide.
Sarah.
Yeah?
Sarah, we're going to go ahead and give a 2-minute warning.
Yeah, I'm almost done.
Okay.
Next. So, and we build a reliability index for those historical data as well, based on glint, ground truths, time of the year, and tide level. Next slide, Rita. Next, please. So, and from that, we were looking at a spatial temporal trend. And here is just an example of 2004 and 2015. We're not necessarily thinking about quantify area per se, but look at presence and absence. And what you can see here is that there's not too much change between those almost 10 years in kelp distribution in this very specific area. Next one, please. And then to finalize, 2004 doesn't make for a baseline. This is very recent history. So what we're looking at is using the British UMRA charts from 1905, which you see a little snapshot of it here, in which they used to draw kelp, which I zoom in the little box in red there. So you can see the little blades of leaves there. And what we're doing is digitizing all those little leaves along the coast of British Columbia and define this as our baseline. So you see the blue dots there represent where kelp occur along some areas of the BC coast. Next slide, please. So here's all the blue and the red dots represent kelp, either at 1905 or 1930. Next slide, please. And if you zoom into that specific area that I was showing to you as a case study, so this is the 2016 unsupervised classification of Worldview 3, and the same area just beside it is where in 1903 there was occurrence of kelp, which we don't see too much difference. It actually, kelp was there since that time and is still in this region, present in this region. And I think I'm finished. Next slide. And yeah. Let me know if you have any questions. You can either email me or Sarah, or any questions right now.
Excellent. Perfect. Thank you, Sarah.
You're welcome.
Awesome. So that we can actually make sure we have a good transition here, we're going to ask if Helen might be ready to give her presentation. And then we'll head into the discussion afterwards, and we'll make sure that we address specific questions for both Mysira and Helen at that point at the beginning of the discussion, if that works for everyone.
That sounds good.
Perfect. So we're going to go ahead and pull up Helen's presentation. And as I mentioned at the beginning, Helen's with the Washington Department of, or Washington State Department of Natural Resources. She's going to be discussing an overview of floating kelp trends and issues in Washington. So give me 1 second, Helen, to find you on here. All right, perfect. And Helen, please take it away.
Okay, can everyone hear me?
Yes.
Yes.
Perfect.
Excellent. Okay, so I work for the Washington Department of Natural Resources, DNR, which is the state steward for aquatic lands. And our group, the Nearshore Habitat Program, monitors a suite of different indicators of nearshore habitat condition, and kelp is one of them. And we're also part of a regional monitoring group called PSEMP. Today, I'm going to start with some really brief background context. Here's Washington State, just south of British Columbia. We have about 5,000 kilometers of shoreline. 10% is along the rocky northern outer coast, which is a classic open coast upwelling driven system. But the majority of nearshore habitat is in Puget Sound, which is a fjord system with large tides, strong currents, and estuarine circulation. And in that area, that's where most of the human development is. So the environmental context for kelp and the stressors are really regionally distinct. And I'll talk more about that. Kelp's found along the marine shoreline of both the outer coast and within Puget Sound, even the inner basins of Puget Sound. These maps, which were based on the shore zone inventory, which we did years ago, shows shorelines with kelp in yellow. And you'll see there's a lot more understory kelp, and that's species that don't form a floating surface canopy. There's widespread concern right now in Puget Sound about losses in kelp, especially bull kelp. And however, the information on trends is really limited. NOAA right now is funding a kelp recovery plan for Puget Sound, and that's an offshoot of the ESA mandated rockfish recovery plan in Puget Sound. And there are also really important links between salmon and kelp that are a high management priority, which Mysira talked about in our region. The first phase of the recovery plan, which we're doing right now, is a literature review. And phase 2, if it's funded next year, will outline actions to be taken to recover kelp. So today I'm going to give you a rapid fire overview of what we're seeing in Washington and talk a little bit about methods. I've seen that you all can digest a lot of information very quickly, so I'm going to take advantage of that. Next slide. So most of this research focuses on 2 species of floating kelp that are found in Washington. This isn't because floating kelp is a priority, it's because it's easy to survey, as we've all talked about. And while survey techniques are well developed, as everyone's discussed, it's important to remember that kelp canopy surveys aren't highly precise, especially in our region, because you have extreme tides, currents, and weather conditions that really affect the amount of visible canopy. Next slide. Oh, and I would like to mention one other thing, which is giant kelp is only present in our system on the outer strait and outer coast. So most of Puget Sound is just bull kelp in terms of floating kelp. So we've surveyed kelp canopies in many ways over the last 2 decades. Aerial photography, airborne multispectral, kayaks, small boats, etc. And towed underwater videography too. I'm going to talk about the longest data set we have that uses consistent methods, because that's where we're learning about long-term trends. And it covers the entire Strait of Juan de Fuca and the outer coast. And it's pretty similar to the CDF and W surveys that we discussed previously. It's not technically sophisticated. It's just highly consistent. It's based on aerial photography of CIR, color infrared photography. It's mapped at 1 to 12,000, and it's collected annually in late summer at low tide. And in our area, low current is really critical, as well as tides and calm sea state, as Mysira showed us. So on the left is an example of our monitoring data at Cape Flattery, and you can see multiple years of surveys overlaid to visualize persistence of kelp in a color ramp. And this is where multiple years of consistent data gets really cool. The yellow represents canopy forming kelp present for less than 5 years, and then you can see it ramping up to purple, which is more than 25 years. So next slide. The graph shows canopy area over time, and the black line is total canopy area, and then it breaks down by species. So blue is bull kelp, and green is giant kelp. And this reminds me of some of the comments Meredith was making about the differences of the two. This data set, as you can see, as we all know, kelp has really high interannual variation. And you can see that in this data set. You can also see that it's stable over this entire monitoring period, meaning no linear trend. Variation is greater in the annual species, bull kelp, which is the blue line. You see it goes up and down a lot. And that's greater than in the perennial. Interestingly, though, at this scale, the two species are positively correlated. So a good year for one species is a good year for the other. We do know they compete at smaller spatial scales, no question about that. So as we all, I think, assume, kelp ecologists have long attributed much of the variation that we see in kelp to climate. And you might immediately notice when you look at this time series that the extreme lows occurred in 1997 and 2014, which were extreme weather events. They always jump out for me. And so next slide, please. We recently quantified the links between canopy area and climate. We finally have a long enough data set to do that using RIMA time series models. And this is published. You can go look it up and get a lot more detail. This is the cross correlation, which summarizes the results. And basically, we saw significant correlations with all three ocean indices we tested. Kelp abundance was greater during the cool, nutrient-rich phases. So a positive correlation with NPGO and a negative correlation with ONI and PDO. And the tightest correlation was as a leading indicator with a lag step of 1 year with NPGO. And so this was helpful for us because it really does confirm what we assume, that kelp abundance is linked to climate. Next slide. We're also really interested in how kelp abundance has changed over time. And so here's an interesting study. We looked at floating kelp area along the strait over a century, and we compared our long-term data set, which I just talked about, to detailed maps that were produced by Rigg in 1911 and 1912. And Rigg's maps were created to identify kelp beds as harvest resources for fertilizer. And this mapping effort occurred all along the West Coast. I'm just using the Washington ones. Oh, by the way, all of the Washington maps are available online. You can go to geo.wa.gov and search for kelp forests, and we have a whole story map of all of this data, which is really cool. So we compared area between the two time periods to ask what's happened over the last century. And we did it at the scale of map indices. So if you look at on the right-hand map, you see those little polygons, the little black numbered polygons. That's the scale at which we compared kelp area over time. Next slide. So what you see here is you see the numbered polygons on the map, and we only compared the polygons that are highlighted in yellow. So we excluded some because we felt there was some uncertainty in the historical data. But of all the ones we compared, you can look up and see a figure where there's a black dot, which represents the area measured in 1911, 1912. And then there's a cloud of orange dots, which represent the 26 annual measures in the last 3 decades. And we didn't do statistics on this because the methodologies violate all sorts of assumptions. But what we did do is we assessed this visually and saw that in most areas, the black dots fall within the cluster of orange dots, which suggests that the historical measure falls within the modern range. There's one notable exception to this pattern. If you look along the eastern edge, you see that the black dots are consistently above the orange dots, and those are the numbers 4.1 to 1.1. This suggests a different pattern in the east, which is the area farthest from oceanic influence and closest to development. So in summary, the strait appears to be pretty stable at a broad scale over the long term. And perhaps there's some decrease in area in the eastern strait in floating kelp. This is not what we think is happening in other areas. So now I'm going to switch, next slide. And I'll switch gears and talk about the inner basin and specifically look at South Puget Sound, which is the most distant from oceanic influence. And what we did here, we don't have a multi-decade monitoring data set here, so we took a different approach. And we all compiled all the observations we could find, which were about 30 data sources, starting in 1855 to present. And I'm wrapping up this study now. The green dots show you when in time the different information sources occurred, and some of them might be a single site, and others might be synoptic mapping of the entire region. So I'm going to show you only the most simplified, next slide, analysis that we did, because there's so many differences in all this data. But here's the most simplified analysis. We divided up all the stretches of shoreline where bull kelp has ever been noted into 29 sites, ranging from 1 to 6 kilometers in shoreline length. And you can see those sites on the map. They're either yellow or light green. And I just did them in different colors so you could see when they're adjacent to each other. And they're also named and shown in the figure on the right. And in this case, it's a very low sensitivity analysis. It's just presence or absence at every site. And you can see every time there's a observation, purple means bull kelp was observed to be present, whereas gray notes that it was observed to be absent. While this is a really rough approach, it allows us to incorporate data sets that aren't comprehensive and ones that only use narrative descriptions, which a lot of the old chart books do. So what you'll see is as you look through this list that's sorted from west to east, is that there's a pattern of decreased persistence, fewer purple dots in west and central, the first two regions, in recent years. And I've drawn a line at 1980, right at 1980, because that was after one of the big mapping efforts. And you can see when you scroll through that, the eastern area, which is the Tacoma Narrows, still has a lot of bull kelp, whereas the two other areas are actually mostly absent. And if you go to the next slide, we can think about this another way, which is you can roll up all these dots and just look at what proportion of observations saw bull kelp either present or absent before and after 1980. And what you see is before 1980, there was a relatively high proportion of observations with bull kelp present, whereas after 1980, the median in west and central is 0, whereas the persistence in the Narrows stays high. The Narrows is an extremely high current, well-mixed area, and so that may be protecting it from the stressors that we're concerned about in other areas. So this is the little work we've done on looking at stressors and losses in the inner basins. Next slide. We hear concern over losses throughout the Salish Sea, so also north of the border, especially in the inner basins such as Georgia Basin. And some of the candidate stressors, here are some of the candidate stressors we think of: elevated temperatures, generally urbanization, anthropogenic nutrients, sedimentation. It's a standard list. Overfishing, community shifts, and two key issues for our region, which are grazing, and one of the big concerns are kelp crabs, and also the non-native alga sargassum, which are both documented to, both have studies that show impacts on kelp. So now I want to quickly change and talk about, with the next slide, recent trends, because we know in North Central California, you all experienced massive losses around 2014 during the extreme weather conditions. So this slide, so I've been very interested in this and talking to Cynthia and asking, well, what happened in Washington during that period? So this map shows areas we monitored with the same long-term aerial photography methods. And what you see is if you start at the coast and the strait on the lower right, the mustard and the purple, they declined dramatically in 2014. But unlike North and Central California, they rebounded in 2015. And then and there's also a fairly spatially complex pattern here. So if you look at Smith and Minor Islands in pink, they also dropped and then increased, but the increases were slightly delayed. And that's also true for Cypress Island in red. There were decreases and then increases. And then finally, Cherry Point is very interesting to us in light green because the recovery there was delayed until 2017, but it did come back. And so we're thinking a lot about what's caused this spatially complex recovery. And we think it might be temperature. Next slide, please, which is a link to the California story. If you look at this AVHRR data that we summarized for this is June and July 2016, you can see how strong the temperature gradients are within the Salish Sea. The strait's really well mixed, the area that recovered really quickly. And if you go up to Cherry Point in the Georgia Basin, you can see that the temps were above 15 degrees. They continued to be above 15 degrees in 2016. Temperatures are just returning to normal now in Puget Sound. And all of this may remind you of the talk you heard by Braden Schiltroff at the last workshop. He's looking specifically at temperature impacts in north of the border on the Georgia Basin side. Okay, next. We have a lot of volunteer kayakers who are great, and their on the groundwork confirms the DNR aerial photography that I just showed you, which is there are a lot of little green sites that as of 2017 are stable. Whereas if you look at the orange sites, you'll see that we still have declining beds in the inner basins. And so we're looking forward to 2018 since we're expecting the high temperatures to continue to ease, and we may see recovery there. Okay, quick summary. What lessons have we learned from aerial methods? Next slide, thank you. Aerial imagery is effective, but it's not precise. Everyone's talked about this. Especially in our region, consistency is critical in methods, tides, currents, and seasons. We contracted with Kevin Britton Simmons to do a study in 2008, and it does a really good job of quantifying effects of tides and currents. You really have to, in our region, you have to control for them as much as possible. Satellite imagery has been problematic for us because we have really narrow data collection windows and narrow beds. So I've read Tom Bell's and Kyle Kavanaugh's work, and I got to say I'm really envious of the number of usable images that they get using Landsat because we get a tiny fraction. And also those 30 meter by 30 meter pixels are much bigger than most of our fringing beds. So we are looking at high resolution satellite data now with a major focus on minimizing the discontinuity in our data stream. And then the last thing I'd like to mention is that we see a major limitation to aerial imagery, which is it only captures canopy forming kelp, which is a tiny fraction of the kelp community. And we want to monitor understory kelp too. So with the last slide, next slide please, I'll mention quickly, we're experimenting with large area surveys using towed underwater videography. And one example that we recently published is at the Elwha River, which is the site of the largest dam removal and restoration project in the US. And you can see in the picture what happened when they removed dams and released 100 years worth of impounded sediment. We had massive losses of kelp and then recovery. And what the figure shows is the USGS dive sites are triangles. They had mini dive sites. And then our towed underwater videography is circles in the central map. And you can see 1 year after the dams were removed, the color coding shows change from initial algal cover. And there were massive losses right around the mouth, and then a gradient of decreasing losses as you got farther away from the mouth. And then the two side panels are our control areas, where you saw very little change. This study, pairing dive and video data, has helped us to think about pairing higher precision and higher detail surveys with lower detail surveys like video, which have greater spatial extent. So that's all I have.
Excellent. Thank you so much, Helen. I also just want to give a really special thank you to all of our presenters today. I think all of your information will be really valuable for this working group moving forward. And it's going to be a good start to our discussion, looking at what can be accomplished in the long term in our region. So thank you so much for all of your time and sharing your expertise with us today. Just to respect people's time, I do want to give us the opportunity to head into our discussion, but I don't want to limit any questions for our last two speakers. So if you do have those questions for them, we can go ahead and address them in the beginning. At this point, I am going to hand it over to Francesca to facilitate the discussion and Q&A session. Now, just as a reminder, we will have notes for all of our speaker presentations and for especially this group discussion and Q&A, but this webinar has also been recorded in full. So if you want to go back and see any of the presentations or listen to any of this part of the discussion, it will be made available to everyone that attended the talk today and all of the working group members, because we covered a lot of information today. So again, thank you so much to all of our presenters. And Francesca, if you're ready, I'm going to go ahead and hand it over to you to facilitate. And now, I mentioned at the beginning, folks can either type their questions still, or you can raise your hand and we can unmute you so that you could verbally ask your question and we can start up a conversation for the discussion. All right.
Okay. Hi, everyone. It's Francesca Coe, and I am the co-chair of the Kelp Recovery Working Group along with Cynthia. And if everyone can look at their GoToWebinar control panel, you'll see that in the top section under attendees, there are several little icons. There's a microphone, there's an exclamation point, there's a question mark, and then there's a little hand. So if folks can raise their hands if they have questions, I will go through and unmute you so you can ask your question. And also still available is the opportunity to put a question in the chat box. So avail yourself of that option. I'm going to now unmute Jan Freewald, who has her hand raised. So, Jan, go ahead.
No, I didn't raise my hand.
Okay.
Sorry.
Maybe, no, that's okay. I will remute you.
There are a couple of questions that are in the chat for Helen's presentation.
Okay. I'm going to go to Mark Carr. Mark?
Yeah, my question is for Helen, and it's the same one that I typed in the chat, which is when you're looking at these spatial patterns of both loss and recovery of bull kelp, do you have data on urchin densities and urchin density trends over that time period?
Thank you, Mark. And Helen, I'm going to unmute you so you can answer.
Mark, that is one of the most interesting topics that I think we talk about is urchin densities. I've had long conversations with Cynthia Catton and Hank Carson, who's the urchin harvest manager up here. And we generally do not see very high urchin densities in many of the areas. So far less than the thresholds. I think last week they were talking about 2, was it a 2 urchin threshold? Last workshop, but here the densities are relatively low, except for in the areas that are closed to urchin harvest. And Hank and I have had long discussions about, do you know of an urchin barren in Washington State? Can we think of an area that we would classify that way? And we really haven't come up with one that really, really obviously fits those criteria. One thing that's interesting is that we have sea otters on the outer coast, and we have human harvesters within, as soon as you get into the Strait of Juan de Fuca. And so perhaps they're controlling the populations. There was really extensive harvest during the late 80s and early 90s, and the population is considered to be a fraction of what it was.
And then I have a question here in the chat box. And so I'm going to read it, and then I'll unmute you, Cynthia, if there's anything else you want to add. What do you think is driving the strong correlation with the 1-year lagged NPGO? And I'm unmuting you, Cynthia.
Yeah, I'm pretty terrible at writing questions on the chat box. You know, trying to keep it concise, but actually also clear. But I was thinking back to the table that you showed, Helen, of the environmental indexes that you were correlating with the canopy abundance, and you said that the 1-year lag with the NPGO index had the highest correlation, and I was thinking about what that would mean. So basically, it's the year before, temperature, nutrients, and all the things that are associated with that index that is promoting or not the the following year's canopy abundance. And do you think that that's driven by the macrocystis or the nereocystis, or did that matter? That play into it?
Okay, let's see. So much to talk about there. I didn't present we actually looked at correlations by species too, and I didn't have time to present those. The correlation is slightly higher in nereocystis, but it is, but for both species, the highest is a 1-year previous lag. And you may also remember that there was no significant correlation with temperature.
Right.
And that's, so I think the main weakness there is that, as I showed in the AVHRR imagery, temperature is really spatially complex in Puget Sound. And the only long-term temperature data that we have is from Race Rocks, and that's in a really well-mixed area. And so I don't know if we would be able, with more spatially explicit temperature data, I don't know if we would see a strong correlation long-term. I think, though, my sense, and I'm far from an expert on climate indices, is that somehow the NPGO is synthesizing both the temperature and the nutrient characteristics, so that you do get a significant relationship between the two variables. And of course, we know that nutrients are really important as well as temperature. The problem we have in Puget Sound is that most of the relationships between nutrients and kelp are empirical, and they rely on temperature in upwelling systems. Most of that work's been done in California. And so we just don't know how consistent that relationship is in Puget Sound between nutrients and temperature. It looks to be consistent, but we can't, we don't have all of that evidence to show that. So summary is it appears that environmental conditions are a leading indicator by 1 year. I think Kavanaugh found that it was closer to 3 years for giant kelp in the area of California he studied. In our region, it was 1 year was the tightest correlation for both species.
Do you think that that's pointing to production, spore production, or like the previous year's conditions? And I think that those indices combine lots of different, are correlated strongly with lots of different environmental factors, and maybe even including precipitation up there. But do you think that indicates to us that we would be thinking about the previous year environmental conditions influencing production of either additional sites for macrocystis or spore production or that sort of thing for the following year?
That's definitely that's my supposition exactly, is that you have two things happening. One is the environmental conditions in the year that they're occurring, and then also you do have lead time with the environmental conditions having an effect over multiple years on the plants.
So next in the queue, I have a question from Rebecca, which is for Meredith, and then I'm going to go to Tom Bell, who has his hand raised. From Rebecca, I'm just going to unmute you, Rebecca. Go ahead, Rebecca.
Hi, Meredith. I really appreciate that you're going to be looking into the other satellites in the future with different spatial resolutions. I was just wondering if you can kind of expand upon your findings when you were comparing the Rapid Eye 5-meter spatial resolution to the CDFW surveys and the Landsat surveys, and you found that the Rapid Eye was overestimating the kelp. Can you expand upon the reasons why for that?
Yeah, so I don't think we know exactly why it is right now. Okay. So when I first started working with the Rapid Eye data, there is no atmospherically corrected product for Rapid Eye like there is for Landsat. So I actually kind of just wanted to try to see if we could, because kelp is a bright target. So I was thinking, maybe we can detect it without doing an atmospheric correction. And so actually the Landsat imagery in that graph is atmospherically corrected and the Rapid Eye imagery is not. And so now I'm working on the atmospheric correction for the Rapid Eye imagery to see if that increases the signal of the kelp, basically. And I have a feeling that it probably is what is causing it to overestimate the kelp canopy so much. But yeah, so basically I just wanted to try and see if it would work without doing an atmospheric correction. And it didn't work.
Yeah, no, I mean, that was, of course you would try that, so yeah. Well, I'll be interested to see and what comes of it once you have done that correction.
Yeah, so it's most of the atmospheric correction programs you have to purchase. So right now, the reason that it's taking so long, atmospheric correction is something that is widely applied to satellite imagery. And so I'm just working on doing the atmospheric correction in a way that is free and not using a program that's expensive. So that's actually, it's actually not that hard, but it's taking some time because of the route that I've decided to take. But I should have those results very soon.
Great, thank you.
Thanks, ladies. Now I'm going to go to Tom Bell. Not sure who's Tom's questions for. So Tom, if you'll go ahead.
Hey, two things. One for Meredith, you might want to just, if you can try like a dark object subtraction. That may be like a good like first order atmospheric correction for the rapid eye.
Right. Yeah.
Which would be.
Yeah, I have tried cloud and shadow, which is relatively straightforward, and it actually didn't work very well. So yeah, to be determined, but dark pixel correction is on my list.
Cool. And then I raised my hand to kind of follow on to the MPGO discussion that was happening before the last question. So yeah, Kyle found in the 2011 paper that there was like a 3-year lag with MPGO. We found depending on the region of California, looking at the 2015 paper, that there was like a 1 to 2 season lag with NPGO. And if you're looking at an annual scale, that probably means more like a 1-year lag than like a present value lag. One interesting part about the MPGO, if you follow on to Parnell's 2010 paper down in San Diego, is when you look at Southern California with macrocystis, the biomass follows the MPGO very closely. And what the MPGO may tell us that sea surface temperature may not. When we look at sea surface temperature, we'll only look at the skin of the surface of the water. And of course, there's probably a breakdown in temperature to nutrient relationships in Washington compared to California. But the MPGO may also relate to processes that we're not seeing with sea surface temperature, like a shoaling of the halocline or nutricline, which could be flooding these shallow reefs with nutrients that we may not be seeing with sea surface temperature alone, which could lead to increased recruitment, which we would see the next year. So that's, and going by the different regions, if you look at Southern California, the MPGO increases the strength of the California current, and actually advects a lot of cold water down into Southern California, which normally would have been advected into the Santa Barbara Channel. And that's why we kind of see this, we think we're seeing this mismatch between the MPGO and the Santa Barbara Channel versus in Southern California. But we also see a strong predictor of the MPGO along Central California as well. It's just a seasonal cycle with waves is on top of that. So it's really complex. We have the new paper that's coming out does a new complete analysis of the MPGO in California, and we think that really is driving a lot of the interannual patterns of kelp canopy biomass we see here. Thanks.
Okay, great. I'm not seeing any other hands. I'm going to unmute you, Cynthia. Earlier in the presentation, when you were talking, or actually not Cynthia, Rebecca. I'm going to unmute you, Rebecca. I'll unmute both of you. This is Francesca. I was wondering, with respect to the aerial surveys, if the folks working on those had thought of or reached out to Lighthawk, because I know they have flight capacity. I don't know what's involved with the capturing of the imagery, but just wanted to offer that as an idea.
Yeah, so when the department began, thank you for that. I'll look into that. So when the department began contracting out the surveys, the way it works with the state is that we develop a request for proposals, and the folks that are interested in putting in a proposal will do that according to whatever the details are in that request for proposal process. So once folks will submit a proposal. They also have a bid that's associated with it. Well, all the proposals are first reviewed and scored based on this predetermined scoring. And any of the applicants that are past a certain scoring range, then they are seen as a responsible bidder. And then the contract will go to whoever is the lowest bidder. So the department can't just pick and choose who they want to do the survey. Yeah, that offers any insight on that process. But thank you. I can look into that, into the Lighthawk.
Yeah, it's a volunteer service, so they probably wouldn't be responding to RFPs, but
Okay.
But you and I can talk about it offline because it might be something that could augment whoever you are hiring as an additional resource.
Yeah, thank you. And that's definitely, I think I may have alluded to it earlier, is that with the department, we are having discussions as, how can we continue this survey? And it could possibly be that, maybe we perform some sort of aerial kelp surveys in some areas, to get that tighter spatial resolution, maybe employ drones and also do satellite imagery for those areas that, we know it works. So definitely some discussion to continue with that.
Rieta, I'm not seeing any other hands, and I did want to unmute. Oh, Mark Carr has another hand up. So I'll go to Mark, and then I was going to say I will go to Josh Russo after that, so Josh can give an update on the Watermen's Alliance efforts at Ocean Cove. But first we'll go to Mark. Go ahead, Mark.
I think I have a short question that may lead to a really long answer. So, and it's for Tom and Meredith and any of the others who are exploring both the remote versus the drone-based survey methods, and what your thoughts are for the North Coast, given the weather conditions and the unpredictability of being able to capture satellite imagery. What do you guys think with respect to exploring drones versus the more remote sampling methods?
I'll let Tom go first since he's actually been flying drones and I haven't done that yet.
Okay. Well, what we've been doing, I've been doing two kind of drone-based operations. One in Santa Barbara, we've been doing them simultaneous to Landsat overpasses and simultaneous to L2ER biomass collection. We've also been doing it in Alaska from boats, mostly in Southeast Alaska around Prince of Wales Island. That obviously is harder to fly drones in Alaska than it is in Southern California. There's obviously pluses and minuses to both. You do get a fair amount of Landsat coverage in Northern California. It's never going to be as good as Southern California, but we do get, at least 6 to 10 cloud-free images per year. The drone imagery, if you have someone there on, that can fly a drone that is living in the area, then it represents a great way to take advantage of good conditions. When you have low wind, even if you have cloud cover, you can still, that's high enough, you can still go and fly a drone and get the imagery you need. Color drones are a lot cheaper, just color camera drones, than a multispectral unit. But we can still get kelp density, at least basically kelp density, because the pixel scale is so small that you have a 4 centimeter pixel. You can basically turn this into ones and zeros and then sum them up into 1 meter, 2 meter blocks and get a good idea of kelp canopy density. Yeah, I think it's a great way forward. Your spatial extent is going to be smaller, but if you have someone on site who can go and fly it when there's good weather, then I think it's gonna we're going to see a lot more drone imagery in the next five years.
Yeah, I also think it kind of depends on your question, just because you can never, or it would be very labor intensive to get as much drone imagery that would cover, for example, an entire, like just one Landsat image. And so I think that it would be really interesting to use a drone to study changes at one site, for example, over time or within a season or something like that, and get higher temporal resolution and much higher spatial resolution, where maybe if we're curious and looking at large scale, oceanographic effects on kelp dynamics, and we won't be able to use drone imagery, but we would need the satellite imagery just because of the spatial limitations of the drone and the labor that is involved in getting that much imagery.
Mark, do you have a follow-up to that?
No, that was helpful. And I guess on the other hand, there's different types of drones. The smaller drones that Tom and I think Meredith that you've been thinking about using, versus the larger drones that could fly the whole coast. It'd be interesting to know, to explore what the relative costs and benefits of those different approaches might be with respect to, how expensive they are, how reliable the data are. And I think, like Tom was hinting at, it really comes down to just how often, how reliable you actually can get reasonable satellite images up and down most of the coast. But to follow up, your response, Meredith, and the previous question, which is maybe it's a blend of the two, is the optimal solution. That, like you're saying, there's benefits of both approaches at different spatial scales, and maybe the thing to do is to think about how to design a sampling program that uses both to complement one another.
Mm-hmm.
Thank you, guys. Before I go to Josh, I see Helen, you have your hand up. You are unmuted.
This is a follow-on question to the discussion that Mark and Tom and everyone was just having, which is we're experimenting with small drones a little, and they seem useful, but yeah, the site scale, the site size seems much smaller, and so then it becomes a complementary technique rather than an alternative to satellite or airborne imagery. And so I'm wondering if you all have thought about what that site size is for the drones in, I don't know if it's kilometers of shoreline, or we're doing small sites, like half a kilometer.
Well, this is Tom. For, like a Phantom 4 Pro DJI drone, you're pretty much limited to about 40 hectares in a flight, assuming conditions are perfect and you're flying at your maximum allowable altitude. So I think that is your limitations, and you're going to have to move from place to place to get more sites. I agree completely with Mark that this and Meredith that this is going to be a complementary method to satellites and aerial overflights in the future. Once FAA regulations are continued to relax on drones, it would be nice to see if we can start exploring fixed wing options instead of quadcopter options, where we can cover larger spatial areas, like Mark kind of hinted at, with drones that may be able to fly larger portions of the coastline. But right now, it's all the FAA regulations are kind of line of sight, so you have to keep an eye on your drone the whole time. So I imagine those will be relaxed in the near future, but we don't know when.
Mark, did you have something to add, or do you have a new question?
Neither. No, that was a great response from you guys. Thanks.
Okay. Then I'm going to go over to Rebecca. You had your hand up.
Yes, I was just going to agree that, it's the beauty of drones is that you can perform those surveys more often. As our CDFW surveys, it's just one time during the year. So the beauty of having those surveys more often is really appealing. But also, as I think Meredith had pointed out earlier, when we found our statewide surveys, it was really interesting because there were some areas that kelp growth looked really great or, normal, and then you kind of just go around the bend a little bit and it had dramatically decreased. So, one thing with the drones is that because it's not may not be as easily to do some sort of statewide survey, we're not going to pick up on those little details.
We have a question from Steve Lonhart. He's asking, what does a multispectral sensor on a drone cost, at least roughly?
Well, go ahead, Tom.
Well, I was just actually flying one yesterday with Kyle over Santa Gita Lagoon and Del Mar. So the redhead sensor, the same one that was Meredith was talking about, I think they're around $4,500. And you could put that, we have it on a Matrice 100 DJI drone, which is another $2,500 to $3,000. I think total, it's about $7,000, 7 to 8. The prices are coming down. I've heard of some people mounting that same sensor on a Phantom DJI drone, which is about $1,500, but I haven't ever seen one in use. So I think about $7,000 to $8,000, but I don't know, give it a year or two and it's probably going to be down to $4,000.
Steve, you're unmuted if you have any follow-up. No? Okay. Then I'm going to, let's see, I'm going to take it over to Josh. And I need you to give us an update from the Watermen's Alliance urchin removal event. Go ahead, Josh. Josh, are you there? Okay, maybe he fell off. Rieta, do we have anything else? I'm not seeing any other hands or any other questions.
At the moment, I don't have anything else to add. I think that it's pretty good timing to wrap up our discussion. I'm really glad we were able to get people's thoughts in towards the end of the webinar and really start to process this information. So thank you all for your active participation, and once more to our presenters for being really amazing in sharing all of your information and knowledge. And I think Josh just sent a message saying he didn't know why he couldn't hear, or why we couldn't hear him. Maybe we could try him one more time.
Sure. Josh, you're unmuted. Go ahead. We still can't hear you if you're talking.
Yeah. Maybe Josh could send a... Oh. What did he say?
Hey, Josh, if you can hear us, would you raise your hand? Because we cannot, okay, you can hear us, but we can't hear you. So Rieta, since we can't hear him, maybe Josh could send sort of a narrative update in an email to the group?
Yeah, I think just a little update, maybe a recap of the event. And he did just send in the chat that the next removal is July 21st and 22nd at Albion. So Josh, maybe if you wouldn't mind just putting together a little blurb, that would be really great, I think, for everyone to know what's been going on and what's coming up. So thank you for sharing that. Great. And I think, yeah, Yeah, so he's raising his hand so he can hear us. But it sounds like we're having some audio issues there. Okay, well, I think, Francesca, that concludes the webinar, unless you have any parting thoughts. I also do want to give a shout out to Jen, who's sitting next to me and advanced all the slides and helped me handle logistics. So thank you so much, Jen, for all of your time today. Much appreciated. And yeah, thanks to everyone. And I think we'll send out a link to a Google Drive document that will have the presentations and will have notes and the recording for the webinar today. So we'll send that out to the group so you'll have access to all of this information.
Great. Thanks, everyone.
Thank you.